What we were doing last time was talking about enzyme activity, and we're going to take that enzyme activity and we're going to start applying it directly to cell physiology, specifically to cellular respiration. So where we got last time, I think, is we did almost everything in the enzyme activity list except for product concentration, if I remember correctly. So I want you guys to tell me, I know it's been a long weekend and you've slept or worked or both since then. So somebody tell me what the concept of saturation has to do with enzyme activity. In your words. Go ahead.
Too much substrate, not enough enzyme.
Too much substrate, not enough enzyme limits the rate, right? So this is basically showing you saturation rate right here. Yeah? Okay. Low concentration of enzyme. If you added enzyme, that's the only way to increase the saturation rate, which isn't something that happens instantaneously, but can happen. Okay. As you increase the substrate concentration, the enzyme catalyzed reaction tends to increase until you hit saturation rate, right? And then that's what this is saying. No matter how much more substrate I added, once I get to saturation, I'm still there. Okay, so now the concepts that people get confused. I want you to tell me substrate specificity. Substrate specificity implies different rates of chemical reactions if I have multiple possible what? Substrates, exactly. So that means that in this situation, right, somebody tell me the difference between the chemical reaction rate with C binding to the enzyme and A binding to the enzyme. Which one has a faster chemical reaction rate? C. Basically, if it binds faster, it drops off faster, you get to use it again, the chemical reaction rate is faster. This basically says that the better the fit of the substrate into a given enzyme, the faster the rate of the chemical reaction. Good? Okay, and then the next concept was enzyme substrate affinity. And enzyme substrate affinity, guys, implies multiple possible what? Enzymes, exactly. We're holding the substrate constant. Let's say it's C, it's its favorite substrate. And we're acknowledging that the shape of enzymes varies both within a person and also between people, right? And since the shape of the enzymes varies, and enzymes are all about shape, structure, interaction, right? Structure, function, interaction. This is an enzyme with a low affinity for the substrate. It just doesn't fit it quite as well, so it takes you longer to get to saturation, right? And this is an enzyme with a higher affinity for the substrate, so you hit saturation more quickly. Now this has to do partially with metabolism between people. So if all things were being equal and she and I were about the same height, about the same age, she might have a faster metabolism than me. And some of that can be accounted for with the fact that her enzymes are different shapes genetically, right? Slight modifications, substitute an amino acid, something like that. They have to still be relatively functional, for instance, cellular respiration enzymes, otherwise one of us would be dead, right? You can't mutate them too much if it's like a biochemical pathway that you really need. But if hers fit better and work better, her metabolism might actually be faster, right? It's one of the ways that you can account for differences in metabolism. Okay, so that is enzyme substrate affinity. Enzyme concentration we went through, you can increase that. Substrate concentration, the only thing that we didn't do was product concentration. So we want to predict what product concentration does to the rate of an enzyme catalyzed reaction. And to do that, let's just make up a fake biochemical pathway. A biochemical pathway is just a series of enzyme catalyzed reactions until you get to the product. So for instance, internally, you don't generally make a large complex molecule by one reaction, right? It's like 25 in a row, right? And if you're in biochem, you have to memorize all damn 25 enzymes. That's not how we're going to do it here. First, we're going to make a fake one. We'll do a real one in just a second. So here's a fake biochemical pathway. I've got A plus B, which is catalyzed by enzyme number 1 to go to C. Enzyme number 2 catalyzes C to dissociate to D and E. And then enzyme number 3 will get me F, which is the product I was interested in. Does that make sense? Okay, so we've got 3 enzymes that are involved here. And so now I've got the product concentration. We're going to go back to the idea of enzymes sort of being shape-specific and having binding sites or active sites for the substrates. We're going to go back to that concept that we looked at before. So if A is shaped like this and B is shaped like this, here's how we're going to picture enzyme 1. It's got a binding site for A and a binding site for B. Yes? That makes sense? Okay. But now I want you to predict for me what will happen as the product concentration starts to increase. First off, I need to tell you that enzymes have sites other than the active site. It's got a site right over here, doesn't compete with the active site. This site right over here we're going to call a regulatory site. Okay, and F fits perfectly into the regulatory site. Here's my question. What do you think F will do when it binds to the regulatory site? Increase the enzyme activity or affinity for the substrates or decrease it? Decrease it, right? Isn't this a mechanism, a negative feedback inhibition mechanism to make sure that you get normal homeostatic levels of F? Does that make sense? So F will feed back to the regulatory site. You can change the shapes of the active sites temporarily by doing this, right? It's not a permanent change, such that A and B, or even just one of them, doesn't fit properly, right? And therefore, you'll slow down the enzyme activity. Why the hell would you want to do that? Well, what if this is the range of product that I want, right? F. So I've got the concentration of F that I'm looking for. Then what happens is when F is low, right, it doesn't statistically find the regulatory site very frequently, right? So the enzyme is on. Are you guys with me? If F is not around, the enzyme is on, and then the product of F increases. And as F increases, it's more likely to find the regulatory site. So it turns the enzyme off, right? And you end up doing this over time. This is a biochemical example of negative feedback being used to maintain homeostasis. Does that make sense? Okay. You guys are quiet. Does that make sense? You got it? Okay. I can tell there are a couple of you who are really, really like facially responsive. Some of you are like, what? And some of you are like flat affect. I can never tell if anything's going on. So I do respond to your faces, especially when they're asleep, and I remember. Okay, so you guys got this? So this concept says that as product concentration increases, what tends to happen to the rate of enzyme catalyzed chemical reaction? Decreases. Okay, so here's a logic question for you. So why does F not feed back to enzyme 3 typically? I mean, it could regulate 3. Why not? Too close. And what would happen if you just regulated 3? I mean, it would still stop the product concentration, right? What? Go ahead.
You know, we'd be inhibiting the last reaction.
Right.
It would still result in CH
all of these. This is a waste of resources, right? And potentially a waste of energy, right? Does that make sense? So generally, it will feed back to somewhere early in the pathway, or if it's a 25-step pathway, it'll feed back to somewhere early, somewhere 5 steps down, and somewhere down again, right? But usually not just one before, because you're wasting all of these intermediate resources and potentially energy if these are energy requiring reactions. Does that make sense? Okay, so now we're going to take that and we're going to apply it because we're going to see examples of it. So this means that you can change enzyme activity. You don't just change enzyme level. So I know we've talked about you can increase the rate or decrease the rate of an enzyme catalyzed chemical reaction by increasing or decreasing the rate of the enzyme. Now, protein synthesis is not instantaneous, and it sure as hell ain't cheap. So sometimes we need the capacity to turn enzymes on or off. We are seeing an example of doing that right here. So there are 2 basic ways of turning enzymes on and off. We just saw one. This one that we just saw is an example of allosteric regulation. Allosteric regulation is a temporary shape change in the active site of the enzyme by binding something somewhere else to a regulatory site. Okay, so this is the same thing that we're just looking at. I've got a modulator. Okay, and notice what it's going to do. It's going to stay the hell away from the active site. It's going to bind to another site. But please note that it will change the shape of the active site because the protein, right, it's all just strings of amino acids, right? And you've got some positively charged amino acids and some negatively charged and some neutral amino acids. What if I just bound something that actually sort of moved the position of one of the negatively charged amino acids? Then the ligand that was trying to bind is looking for a negatively charged amino acid, doesn't find it, and it decreases the affinity, right, of the substrate. That make sense to you guys? That's temporary, but it can, of course, slow down the chemical reaction by changing the rate, right, or by changing the shape. So this is usually, right here, a loose reversible binding, meaning hydrogen bonding or ionic bonding, usually not covalent bonding, something that attaches and falls off and attaches and falls off and attaches and falls off. So you can change the activity of the enzyme temporarily. This is more efficient, of course, than making it when you need it and not making it when you don't need it, right? So just changing it. It's present and you can change it. So let's look at a couple of situations in the body in which this is used. The modulators that we're looking at right there, often in the body, those are called cofactors or coenzymes. And what they do is they change the rate of the chemical reaction by temporarily binding to a regulatory site, and you have run into those very often as vitamins and minerals. So here's the interesting thing. Vitamins and minerals are not usually used in large quantities as structural components of parts of your body, but you need them in order to make these structural components. So what the hell are you using them for? Why do you need zinc? Why do you need phosphorus? Why do you need vitamin D? Why do you need those things? Because generally what those are doing is they are actually binding to a regulatory site and activating some part of the biochemical pathway. Does that make sense? And if you don't have adequate amounts of them, then if you have 0 of them, you can die. If you don't have adequate amounts of them, it will affect the metabolic activity of that pathway. A good example is someone like me who tends to be pretty anemic. What happens to my energy level, my red blood cell production, when I don't get adequate levels of iron, is that I'm completely exhausted. But it's not just iron being part of my red blood cells, it's also iron being a cofactor in some of my biochemical pathways. It is needed to activate them. And without it, they won't go very efficiently, right? So sometimes you can just add back that vitamin or mineral and you can increase the rate of the chemical reaction. Some people who have really, really, really crappy diets, if you take a multivitamin, your metabolism will actually increase. But most of us don't have really super crappy diets here in the United States. I mean, we do, but not lack of nutrients, just like too much fat and salt. So what's the difference between a vitamin and a mineral? FYI, don't know if you care. A mineral is inorganic. You could get it by licking a rock, as long as you could find the right rock. And a vitamin is organic. You have to eat a living thing or a product of a living thing in order to get it. But they're both needed in trace quantities. Like it's not like an amino acid that you need large quantities in order to make proteins. Okay, so there is one other way to regulate enzyme activity that we're going to run into, and you probably run into before, although I don't know if you thought about it this way. And this one is harder to do. It's usually slower. It's usually more energetically expensive. It's very likely to happen intracellular, not in the extracellular fluid. And this is called covalent regulation. So give me a second, and it will make sense, I promise. It just doesn't initially. So notice here that this was allosteric regulation. All we were trying to do was to change the shape of the active site and make it reversible, right? We're going to do the same thing with covalent regulation. We're going to change the shape of the active site, yes? Okay, and it's going to be reversible. But this is more likely to be energetically expensive, and it tends to take a longer time. And the change tends to be permanent until you specifically undo it. So it's not like it's going to bind and then fall off, and then something else binds and falls off. So it's called covalent regulation. If you're talking about covalent bonds versus hydrogen bonds versus ionic bonds, covalent bonds are stronger, they're more difficult to form, they're more difficult to break. You guys with me on that? Okay, so here's what I've got. I've got this enzyme right here. What I want to do, let's say it's in the off position, the enzyme isn't functional or fitting the substrates. What I'm going to do is I'm going to take a chemical group from something else, and I'm going to use a friend enzyme. In order to do covalent regulation, you need 2 buddies, okay? I'm going to use a friend enzyme to yank that chemical group off and covalently bond it to me, and that's how you change the shape of the active site. Here's an example for you. that you've probably run into before. Sometimes we use ATP not just as an energy molecule, we use it as a source of phosphate. And we take the phosphate off of ATP and we phosphorylate another enzyme, right? And we call that enzyme, this one right here, if this were ATP, this would be called a phosphatase. No, sorry, a kinase. Kinase, this one's a kinase. Kinase adds the phosphate, and then phosphatase takes it back off. Does that make sense? Okay. So what we are doing is the same thing. We are just doing a relatively permanent change, right? If we phosphorylate something, it's called a kinase, and let's say we just turned it on. How do we turn it off? Turn it off. We take it back off, right? But notice you're trying to accomplish the same thing. Do you see that this is going to be a little slower? Also a little longer lasting, right? So this stays in this state until there is an enzyme that is actually going to take that phosphate off, right? Why would you want to do this? Well, there are enzymes that you have inside the cell that you want on under certain circumstances and off under certain circumstances, right? And we're not just waiting for something to just bind to the regulatory site. We're actually doing it on purpose, turning these on and off. Everybody good? Go ahead.
So you said it requires two enzymes, but what enzyme A forces enzyme N to chemical group together?
Yeah, that's what it does. Enzyme A, yes, forces that chemical group onto the enzyme. And by the way, sometimes enzymes can do their reverse too. So I'm saying that it requires both kinase and a phosphatase. Some enzymes can actually do the reverse, but we're not going to complicate things by talking about that. Everybody okay with that? Okay, so now the big concept here is simply that enzymes are not produced and then always active. We actually have the capacity to regulate them either temporarily or more permanently, right? So we don't just have the capacity to make more when we need it. So now we're going to take that and we're going to apply it to metabolic pathways. This is a fake metabolic pathway. Metabolic pathways are going to involve enzymes, right? Sometimes 50, 60, 70 different enzymes, okay? It's going to be a series of enzyme catalyzed reactions in which the intermediate is used to get the next ones, to get the next ones, to get the next ones. Arguably in you, the most important biochemical pathway is cellular respiration. And I know you've learned it before, and if you're anything like me, you learned it just enough to get through the test and hoped nobody would ever talk about it again. Okay? I like had Teflon brain to learning cellular respiration, which is ridiculous because I'm a molecular biologist. But I was like, oh my God, please shut up. I don't ever want to hear about that again. But my objective today will be to make it relevant enough that you actually want to retain it and to understand what it has to do with human physiology. So cellular respiration is, of course, the mechanism by which you take nutrients, glucose included, but not limited to, and break them down to ultimately get ATP and generally produce carbon dioxide as a byproduct, right? And that is arguably in you the most important biochemical pathway. But you're not as cool as a plant, because I want to tell you that what a plant does is take sunlight and carbon dioxide and makes the damn sugar in the first place, right? And then, I don't know if you guys remember this, but plants can also do cellular respiration, right? So plants are way cooler than us, and as we destroy the whole damn planet, they're going to outlive us, okay? They're going to come back because they don't really need you, FYI. They also make carbon dioxide. Now, why the hell didn't I study plants? Because when I poked them, they didn't wiggle, so I just ended up studying animals. I was like, this is super interesting theoretically, but like, I agree that they're cooler than me, but they don't wiggle, and mammals do. Okay, so we're going to talk about stages of cellular respiration. And so you're going to have to learn cellular respiration, but you're going to have to learn it as applied to human physiology, okay? So I'm going to try to make you give a shit. And then also, I don't care whether you balance it, because if you try to balance it and do it wrong, I have to count it wrong. So we're going to work on the unbalanced summary equation of aerobic cellular respiration. So you know it. This is the one where you don't have to keep up with the NADs and the inorganic phosphates and all, because you know you're going to do it wrong, right? You're like, I know there's like something in there. There's like 36 or something. So here is the detail that you need to know for me, but you're going to need to apply it. So we're going to start with the brain's favorite sugar. This is not the only sugar that you have the capacity to burn, okay? And in addition, although you won't have to memorize the chemical equation, you can also break down fats and proteins to do this. But we're going to start with glucose, which is the brain's favorite sugar. And to get the most out of it, you're going to need oxygen. And then you are going to produce this as a byproduct, which can become problematic. We know it can screw up your pH if you don't get rid of it. This is called metabolic water, which is really just water. And it just mixes with the rest of your water. Not particularly important to you. Not particularly important to you. If you're a desert animal, interestingly, desert rats reclaim all of their metabolic water. It's one of their mechanisms, evolved mechanisms for living in the desert. You don't really care about this water. And then the entire point of the thing is ATP. And the reason the entire point of the thing, ATP, is important right now is because I already told you that you use 60% of that just to do what? Yeah, to maintain homeostasis. So in the absence of this, you are a flesh-colored pile of goo. You are entropy. So we are going to need this cellular respiration. So before we go any further, to convince you that you should give a crap about this, just this once, I want to tell you that my argument is that this equation right here is the entire reason that you eat, the entire reason that you breathe, and also the entire reason that you have a cardiovascular system. So you eat primarily to get large organic molecules that you can actually use catabolic processes to get energy out of, right? It doesn't have to be this one, but it has to be one that you can use. You breathe just as much to get this in as to get that out. And as a matter of fact, this level becomes problematic more quickly than that level, okay? And you have a cardiovascular system because you are too big. You're not microscopic. You can absorb glucose here and absorb oxygen here. And in order to get everything to your cells, you're actually going to have all of these. Okay, so with that in mind, let's start talking about the stages of cellular respiration. Okay, so there are 3 stages of cellular respiration with an asterisk. If you're a biochemist, you're going to say there's 3 plus the linking reaction, and then you can break it down a little further. So there is, glycolysis, right? There is Krebs, which you have to have the linking reaction before Krebs, and then there's oxphos. So I'm going to show you how freaking complicated these are, and then I'm going to simplify them to the point that you should be able to explain them to your mom, to your spouse who hasn't had physiology, or to your very bright 10-year-old, right? And if you can't do that, then you don't know it well enough to apply it to physiology. So before we get into the simple way, I want to show you the complex way. If you take biochemistry or get a chemistry degree, did you do biochem when you got your chemistry degree? Okay, then your evil biochem professor will make you memorize this is actually glycolysis. It's a series of 10 enzyme catalyzed reactions, each of which has an enzyme associated with it. You have to draw all of these bastards correctly all the time and get the enzymes, right? I don't know how I did that. As a matter of fact, I think I must have just been smarter then. So that is glycolysis. That's just the first step. Here's Krebs. But you go through this twice, right? You go through Krebs twice. 8 enzymes catalyze reactions that you go through twice. And here is the electron transport chain, also called oxidative phosphorylation. So let's take a breath and realize that you could be learning this in far more detail, and make sure you pay attention when I'm teaching it to you simply, okay? So what we are going to do instead is look at the big picture and how it relates to human physiology. Okay, so please note that what you're doing in this figure is you're taking glucose, and we'll follow glucose first, right? We're sending it through glycolysis and getting the output, right? We're sending that through Krebs and getting the output, and we're sending that through oxphos and getting the output, okay? So that is the kind of detail. So let's talk about glycolysis. Glycolysis, the name is telling you what it's doing. It's glycolysis. So what is it doing? It is breaking down sugars. It doesn't have to be glucose, right? You can do glycolysis with other things, but it has to be a simple sugar. If it's not a simple sugar, you'll break it down before you do glycolysis with it. Glycolysis, by the way, is older than eukaryotic cells. It happens in prokaryotes, meaning it precedes the existence of a mitochondria, okay? Precedes the existence of a mitochondria. And you can do it in the absence of oxygen because it actually predates high levels of oxygen on this planet. You can do glycolysis in anaerobic conditions, okay? So I know you memorize, for those of you who are memorizers, that cellular respiration happens in the mitochondria, but that's only kind of true. Like the second two steps of cellular respiration happen in the mitochondria. The first step happens in the cytoplasm of cytosol. Okay, this is where your carbs start breakdown. It does not require oxygen. It can, and it does occur in anaerobic conditions. For instance, in your skeletal muscle, if you are working your skeletal muscles harder than you are supplying oxygen to them, then they will start going through anaerobic cellular respiration, and they will start generating lactic acid as a byproduct. That's just basically going through glycolysis over and over again because you can't go any further because you don't have any oxygen. Okay, so if oxygen is present, then you get to go on to the action stages. If oxygen is present, this byproduct of glycolysis, which is called pyruvate or pyruvic acid, and if you care, C6H12O6 is just broken into 2 3-carbon molecules, right? And then you send those through the rest. That's pyruvate or pyruvic acid. So if oxygen is present, you keep going. If oxygen isn't present, you either stop there or kick yourself back through glycolysis. It costs a little energy to kick yourself back through. Okay, so if oxygen is not present, then all you can do is stay here. If you're you and you're a mammal, you kick yourself back through glycolysis and you generate lactic acid as a byproduct. But if you were a yeast, you'd kick yourself back through glycolysis and generate ethanol as a byproduct. You could just sit around and hold your breath and get drunk, right? Wouldn't that be amazing? You didn't know that yeast and plants were cooler than you. All right, but you can't. You just get lactic acid, which just causes your muscles to be fatigued. Okay, so what do you get out of this? What you get out of this is over on this side. So what we did is we used glucose in glycolysis. So what we're going to do is we're going to keep track of where all of these are used and all of these are generated. Okay, so glycolysis is where you use the glucose. Did I use any oxygen? Nope. Did I generate any carbon dioxide? I don't generate carbon dioxide there. I didn't generate water there. I do generate ATP, and generally they say that you generate 2. What you do is really use 2 to generate 4, so you get 2 net, right? So you get 2 ATPs from glycolysis. Okay, so that's not impressive. 2 ATPs, whatever, okay? But you use the byproducts of glycolysis to get you into this next step. Now, if you were a biochemist, you would disagree with me. I am going to call both of these collectively, I'm going to refer to as Krebs, okay? Krebs is really the linking step plus Krebs. So notice that we are going to get carbon dioxide out of both of them, and we're going to get some more ATP. So I've got pyruvate. It has to be transformed into this acetyl coenzyme A. The only thing interesting about that is that, listen, it's a coenzyme. We just learned what coenzymes do, right? They actually sit and change the shape of other enzymes to increase their activity, right? So you don't have to memorize the structure of acetyl coenzyme A or anything. So pyruvate gets transformed into that, and then you send it through the Krebs cycle twice. And Krebs is also not super impressive, but it does generate 0 to 2 ATPs, and it does generate carbon dioxide, so that's important. So we're going to go through this one more time. Did we use any more sugar? No, we just used the breakdown products from the first molecule of sugar. Did we use oxygen? Yeah, we use oxygen in Krebs. Did we generate carbon dioxide? Yeah, we did generate carbon dioxide, so we're keeping track of where all of this stuff comes from. Did we generate any metabolic water? No. And we potentially generated between 0 and 2 molecules of ATP from Krebs. And why do I say 0 to 2? It's because there's a couple of different ways that you can go about it. One way you don't generate any, and the other way you generate 2. And sometimes one pathway is available and the other one isn't. So it's between 0 and 2 with Krebs, but still not impressive. So you still only got 4. So why the hell did you go through all this trouble? Because what I needed down here was coenzyme, again, coenzyme 2H. And coenzyme 2H can be broken down to boatloads of ATP, a little bit of metabolic water, and it requires oxygen. So that process is OXPHOS, oxidative phosphorylation, which should tell you immediately that you actually need oxygen for it. Same questions again. Did we use any more sugar? No. Did we use oxygen again? Okay. Did we generate any more carbon dioxide? We did not generate any more carbon dioxide. That all comes from the middle part. Did we generate metabolic water? Yeah, we generated metabolic water. And here's the impressive part. The reason you try to do all of this is because by the time you get finished with OXPHOS, OXPHOS generates 34 ATPs. That's why you have to go through all this crap. So you have to get to that intermediate product so that you can send it through oxidative phosphorylation. That means that in the presence of oxygen, a single molecule of glucose, one time through cellular respiration, can generate what range? 36 to 38 ATPs. Okay, question for you now. In the absence of oxygen, a single molecule, one time through, can generate how many ATPs? 2, one time through, right? Okay, so I want to bring this back in for a second, and I want to apply it to human physiology. And I want to think about a really energetically expensive cell like a cardiac muscle cell. Okay, they're super energetically expensive. Okay, cardiac muscle cell has almost 0 capacity to do anaerobic cellular respiration. It can't do what skeletal muscle does. So now I want to think about a cardiac muscle cell in an untreated type 1 diabetic. Okay, cardiac muscle cell in an untreated type 1 diabetic. So what's the relationship of type 1 diabetes to this? Glucose. What does type 1 diabetic has low levels of insulin, right? Untreated, way too low. This happens inside the cell. Can we agree? In the absence of insulin, can you get that sugar inside the cell? You can't get the sugar inside the cell, right? So what happens? Why are type 1 diabetics, for instance, at much higher risk of heart disease and heart attacks than the rest of us? Because even if the blood is going there, correct? Then the sugar can't get in, and then you cannot get ATP, and you can't maintain homeostasis in that cardiac muscle cell, and therefore no contraction occurs. Does that make sense? Okay, so this is directly related to the story that you're telling about this. So what are the options in that situation? And is it only cardiac muscle? First off, why is cardiac muscle a big deal to kill off? Amitotic, right? What'd you say?
It doesn't really grow back.
No, it doesn't grow back. Cardiac muscle cells and neurons, almost, there's an asterisk because there are a couple instances in which they've made them, almost completely amitotic. You lose it, it's gone, right? Okay, so a couple things. What the hell could you do? Well, of course, you could treat the type 1 diabetic to make sure that they had adequate levels of insulin. Or you could change the diet dramatically. Do you see that there are other mechanisms of actually getting ATP in the cells that are not insulin dependent? Do you know that the Atkins diet, which is low levels of carbohydrates and pretty damn high levels of protein and fat, was actually developed for type 1 diabetics? Do you see what you're doing? You're getting around your insulin issue by actually using other sources of ATP in cells. Does that make sense to you guys? So this is all directly related to human physiology. And so that's why I want to try to convince you that like this particular story is a story that doesn't go away and ends up becoming relevant in every body system that you go into. Go ahead.
I have 2 friends with type 1,
Uh-huh.
and they have little devices with
Yeah,
them.
uh-huh.
Are they like insulin pumps?
Yeah,
Or
insulin.
How does that work if the insulin isn't getting into like, I don't understand how it works with the...
Okay, so, well, we'll talk about insulin pumps in excruciating detail a little bit later. Insulin pumps are for type 1 diabetics who have generally been very, very successful at regulating their blood sugar by other mechanisms because they're really, really expensive and they don't give them out like candy. What they do is there's 2 brands of insulin pumps. One of them actually has 2 injection sites. One injection site that's sort of consistently testing the blood glucose. Instead of just every hour, it will do it regularly. And then assessing the blood glucose and then figuring out that little thing has insulin in it, how much to dump into the subcutaneous fluid, which then ends up in the bloodstream. Does that make sense? Okay. And the other one, you prick your finger and then you put the glucose level in and then it actually dumps it in. So that's what it's doing. It's actually like taking your negative feedback, your reflex arc, right, and putting half of it external because your reflex arc was broken. Go ahead.
So it's an integration system.
It is an integration system. It's an external integration system is what it is, right? And the pump itself is actually acting as the integration center, right? And it's actually got everything but the last part, right, in it, right? It's taking the information in, it's deciding what to do, it's putting the information back out, but you still have to depend on your cells as effectors. Does that make sense? Okay, so hopefully I've convinced you that cellular respiration is relevant, even if you still don't like it. Hopefully you see that it's relevant. That was my objective. So what do you need to do before your first test? Which is coming up in like 2.5 weeks, something like that. Okay, so you do have to know this, yes? You have to know the unbalanced summary equation for cellular respiration. If you balance it right, or else I will count it wrong, because it's not true if you tell me the wrong thing. This is unbalanced, right? You also have to, of course, know where everything is used and where everything is generated, right? And then I want you to work on 3 sentences or fewer of describing the relevance of cellular respiration to human physiology. And don't say you would die without it, because you literally knew that before we had this conversation, right? So find some specific application, use the word homeostasis, listen when we use examples of when we use ATP. Listen for something specific so that you can actually put something in there that shows me that you were conscious.
Are you recording?
Huh? Yeah, it's recording. Yeah, so you put that in there. So that's going to be one of your questions. It's going to be like a mini essay question, and you exactly like that. Exactly expect that. So hopefully I will develop some credibility with you when that shows up on the first test exactly that way, because I'm telling you, I want you to be thinking about this. So basically what I want you to do is use this in the back of your mind and start listening as we go through. And when we use ATP, you're like, oh, cellular respiration. Oh, cellular respiration. Oh. And then you start adding it, right? 3 sentences or fewer, because I know some of you guys will write me a damn novel, right? I ain't got all day. Okay, so now what I want to talk about is carbohydrates. Okay, so it is pretty damn obvious that carbohydrates are important, but they are not the only thing that you can burn. We talked about that, yes? Okay. However, your brain is picky. Your brain prefers glucose above all other nutrients, okay? And it doesn't switch very easily off of glucose. So we must have glucose storing mechanisms and retrieval mechanisms, yes? Okay. Now your brain can burn other things, but it's going to be pissed when you try to make it. Has anybody ever done Atkins or a high protein diet? Oh my hell. You get headaches. I lose like 25 IQ points. My husband likes these high protein diets. He's always like trying to buff up. And so I do most of the cooking, not because I'm a woman, because I'm a way better cook than he is. So like we all end up doing these damn high protein diets, and all of a sudden I'm like, why the hell am I pissed off and stupid? It's because my brain is mad that I am not giving it any glucose. However, you do lose like 15 pounds, like really, really quickly. It's not particularly healthy if you've got other kinds of problems, especially if you've got liver or kidney problems. But let's talk about your plan B, your plan C for sugar. So glucose is important. Your brain is the most important organ in your body. Just ask it, right? It's the one that can rate things, and it says me first. Okay, so here's the deal. You have to have mechanisms to store and retrieve glucose, right? So glucose is what we want. We can use other things, but we don't like to. And we use glucose, of course, for glycolysis, right? That's a term that you know. You guys with me? And then glycolysis with a couple other things will get you ATP, which is the point. So we know that already, okay? So what happens when you have eaten plenty of glucose and you've used it everywhere you need and your brain is totally happy. And you are in times of glucose plenty, like after Halloween, right? You're in times of glucose plenty. That is the time in which you should make a deposit, correct? Into your glucose savings account. And the deposit, when you actually store it, does anybody know what you store glucose as? You store it as glycogen, and the primary place you store it is where? The liver, okay? You also store it in your skeletal muscles, but those are like piggy banks. They don't share with other cells, okay? And the process of doing this is called Glycogenesis. So just look at the name, Genesis, glycogen. It's the beginning of glycogen, yeah? Okay, I got a concept check question for you. What if I told you that either insulin or glucagon is the trigger for glycogenesis? You tell me which one. Think about it. Insulin is the trigger for glycogen. So now with the diabetic, not only do you have problems taking in when you eat, but you also have problems with storage of glycogen. Do you see what I mean? Then in between meals, they don't have the glycogen stores that someone who's treated properly has. So they are going to have booms and crashes. Do you see that? Okay, so this is relevant as well. Okay, so times are plenty, you do storage into glycogen. And now I am between meals, or I'm so damn sick that I can't eat, or it's the first trimester of pregnancy. How the hell do any of us stay alive in the first trimester of pregnancy? Most of it is glycogen and another hormone called cortisol, because sometimes you can't eat anything, like just crackers, like 3 crackers a day for 6 weeks. So how do you keep your brain alive during that time? We have to be able to make withdrawals when times are tough, right? And that withdrawal is called glycogenolysis. Lysis of what? Glycogen. Do you see? Okay, store, and this is a withdrawal. And I'll bet you can tell me one hormone that triggers glycogenolysis. Insulin triggers storage, then glucagon. You can eat it when your glucose is gone. Triggers glycogenolysis. You got it? Okay. Everybody okay with that? Okay. You can feel that. That's when I was telling you that you can feel like when you're starving, stupid and angry, still don't have time to eat, and all of a sudden you're like, oh, it's fine. I'm fine. That's that. You just dumped a whole bunch of glucose into your bloodstream, right? But you're going to run out of that, right? Glycogen is not an infinite storage vesicle, right? So you do that. Okay, so plan A, to keep your brain alive, would be eat glucose, yeah? Okay. Plan B is to use stored glucose. And now we're having a really, really rough time. Plan C, what happens when I am starving? When I am literally starving, not metaphorically starving. When I can't eat, or I won't eat, or I'm on a desert island and there is no sugar for me to eat anywhere, does my brain go, it was a good run, guess we're done? No, your brain does not. Your brain goes to a distant plan C, and the distant plan C is to know that if you can't get any sugar anywhere, the brain will be pissed off, but it will burn other things and make them into glucose. So here's what happens. If you are starving or if you won't feed yourself any glucose for whatever reason, what you will do is you will take amino acids, let's say in the starvation condition, and you will transform those into glucose, and then you will do glycolysis. Why not just burn fat in the brain or burn proteins in the brain? Because the brain has almost zero capacity to burn those. The rest of your body would be burning fat, right? It's like other cells can burn fat, but the brain's like, nope, you give me some glucose, damn it, I told you what I like. So it takes amino acids, and it goes through this process. This process is called Gluconeogenesis. Do you see the meaning of the word? The beginning of new glucose. Yeah? Where do you think in a starving person it's going to get these amino acids? Muscle mass, yes. You start to eat yourself. Yep. It's how you keep yourself alive. And you will consume your muscles, and then you'll start consuming your bone mass to feed your brain. Go ahead.
What happens when they begin to blow?
What's that mean?
There's like, I can't remember what it's called, but in places where they are super malnourished. What?
Oh, when their abdomen starts to,
Yeah.
oh, that's the buildup of okay, so this we will see when we start to burn proteins. There are byproducts of this that tend to be really acidic and also cause a tremendous amount of water retention. So this is not without its risks. Okay, so you're trying to keep the brain, it causes ascites, which is when the abdomen gets really distended, but the muscle's pretty much gone everywhere. It's a buildup of fluid because of the byproducts of this process, which is gluconeogenesis. Go ahead.
In this process, once to the point you're starving, you haven't eaten, you're malnourished, when you die, you start eating a whole ton all at once.
Would you die if you started eating a whole ton?
It's happened before, like.
Yeah, it has happened, but I don't know what the fizz is of that. That's a good question.
You die, you just don't.
I like that question. I won't remember to look it up, but if you send it to me, maybe I'll look it up because I'm curious. Or if you look it up, you can teach me, because that's like an interesting question. Yeah, when someone has been starving, you have to like give them food really, really slowly, but I don't know why. I don't know why. I know there's some simple fizz behind that.
It probably diverts resources from your digestive system in order to keep you living. So when you do eat,
And then your digestive system is just absolutely not
I
prepared
mean,
for it.
not absolutely, but pretty close.
Probably cause severe diarrhea, which would then cause its own kind of other problems. I don't know. It's a curious question. I don't know the answer to it. If you remind me and email me, I might look it up for you, and because I think it's probably relevant. Okay, so this happens, gluconeogenesis right here. It happens primarily in your liver. And also your kidneys remember how to do it, interestingly, although they don't do it except when the liver is completely taxed with other things. That's called gluconeogenesis. And gluconeogenesis right here. Oh wait, that's glycogenesis. That's the other one. Gluconeogenesis right here. Here's gluconeogenesis. Produces some byproducts that are going to be problematic. So when you take glucose and actually make it into, you're going to end up with a buildup of ketone bodies, which can mess up the acidity of your body. So the only reason I'm telling you that is because sometimes people lightheartedly take on super high protein, low carb diets. It's really hard on your liver and your kidneys to do those. So as you decide to do those, you need to realize that you are taxing your metabolism in a way that will reveal whether you have liver or kidney problems. So you need to take in tons of water when you're doing high protein diets because the waste products are actually flushed out through your kidneys. So we will talk about that when we get to protein metabolism, which is next because it's related to protein metabolism. You can take proteins and burn them directly if you're not a brain. If you're a brain, you have to make them into glucose and burn them, and they have different byproducts from those metabolisms. Okay, so FYI, these terms all are similar enough that you need to be able to find a way to make sure that you can distinguish between them. But please note that the words are actually trying to tell you what they mean. Glycogenesis, the beginning of glycogen. Glycogenolysis, right? The breakdown of glycogen. Glycolysis, the breakdown of not glycogen, but sugars, right? Okay. Gluconeogenesis, the beginning of new glucose. On a multiple choice question, I'm going to put all 5 of them every single time, okay? Because you need to know them. You need to know them separate from one another. And not just for now, you will actually see these all semester long, because as we come back to insulin and glucagon and cortisol, we will say, causes gluconeogenesis or glycogenesis or glycogenolysis. And you need to know what the words mean. So you gotta like digest those things. Okay, I'll see you guys on Thursday. Yeah.
Refeeding syndrome.
What?
Refeeding syndrome.
Okay, I'll look up refeeding syndrome.